Hello, everyone. This is Brooke Stoutjesdyk, and I'm going to tell you about my study that I did with determining the anti-cancer components of dandelion seed extract. So, a little introduction. Um, Obviously, everyone knows that cancer is a huge issue. It's actually the second leading cause of death in the United States. And in 2021, approximately 1.9 million new cancer cases were diagnosed, and about 600,000 cancer related deaths occurred as well. Um, so, Terataxum officinale, which is commonly known as dandelion, has been used in herbal medicine for centuries due to its anti inflammatory and antioxidant components. It also can be traced back to the glacial and interglacial times of Europe and is accepted to have colonized the Americas in the pre uh, Paleocene period. So it's been around for a long time. Obviously, we know it as a weed. Um, and so we tried to look into the photochemistry of dandelion extract and determine its anti-cancer components based on the phytochemistry. And phytochemistry is the discovery of bioactive nutrients, plant chemicals that may provide desirable health benefits to reduce the risk of major chronic diseases. So basically, using uh, components found in plants to cure some sort of disease. Um, I set up my research to research the phytochemical properties of dandelion and discover the anti-cancer effects that it could have. Um, so in the lab that I work with at UCO, they have studied the anti-cancer effects of dandelion on HeLa cells, bladder carcinoma, and Ramos cells. So I wanted to expand on that research and look at HeLa cells as well as HDF cells, which are fibroblast cells naturally occurring in the body. The hypothesis and the goals of this study. So the goal of the study was to determine the concentration of dandelion seed extract needed to effectively destroy cancer cells while doing minimal harm to healthy cells. My null hypothesis, with this means that there would be no difference um, in HeLa and HDF cell death based on the amount of dandelion extract in each sample. The alternate hypothesis I came up with is that there will be consistent 100% HeLa cell death and less than 50% HDF cell death when the cells are exposed to a concentration of dandelion seed extract at 1% concentration. So to do this, I first started by culturing HDF and HeLa cells in what's called DMEM, which is a media we use under standard conditions, which is 37 degrees Celsius, 5% CO2, 90% humidity inside an incubator. Once the cells were about 80 to 90% confluent, we then subcultured them into a 96 well plate, and different concentrations of dandelion seed extracts were added to each column of the cells. And these concentrations generally went anywhere from 0 to 2% concentration of the dandelion seed extract. But most of the time we looked at between 0 and 1. We then let the treatment sit for 24 hours. And then we performed a Presto Blue viability assay. And this is a test that we run that gives us what's called a viability percentage. And this tells us what percent of the cells are still alive after they were exposed to a certain amount of dandelion extract. And this was repeated 3 times for HDF cells and 3 times for HeLa cells. Here are the results we got. You can see on one side we have the concentration of the dandelion seed extract, and from anywhere from 0 to 2%. Most of the time it went from 0 to 0.8%. That was because the first time we ran the test on HeLa cells, we were getting 100% cell death after 1%. So we decided to zoom in and do between 0 and 0.8 so we can get a more accurate reading of when the cells will specifically die. And then on the other side, there's the viability percent, which tells us how much percent of the cells are still alive after they're exposed to the dandelion extract. And you get, as you can see, we did have some numbers that are over 100%. And that's because the zero, we didn't give it any dandelion concentration. That is considered the control group, excuse me. And that is set at 100%. So the if any of the cells that were exposed to the small amounts of dandelion seed extract grew better than the control group, there would be more cells alive than in the control group, and thus getting over 100%. But that's not an unexpected result. So here are my 3 tests for Gila. As you can see, we're getting most of them dying off around the 0.6, 0.8 concentrations. And then we did the same tests with HGF, this time only going from 0 to 0.8% of concentration being the dandelion seed extract. And as you can see, most of the cells, while they were surviving long enough in the middle range from like 0.3 to 0.5%, they were also dying off around the 0.6 and 0.8% concentrations. So while we were doing the early testing, the data appeared to be showing that there was a significant difference between the effects of the seed extract on the HeLa cells versus the HDF cells. And as I showed in the previous slide, this is because you could see that in the middle, especially in the earlier stages, we were getting pretty much no cell death until the 0.6 concentration. And if you look back up at the HeLa cells, we were getting, they're about 50% dead in the same place that they were about 100% alive in HDF. So this looked like promising results. However, we ran an ANOVA test once all the data was corrected, averaging all the data out and then running an ANOVA test to determine the statistical value of this data. And we got a p-value of 0.068. And this is it's not a good p-value. Basically, it indicates that the data collected, it does not have statistical significance, which means that the difference between that the dandelion extract had on the HDF cells versus the HeLa cells were not significant enough. There was not enough variation for it to have any significance. And that's very prevalent when you put it all into a graph, and you can see the blue line is the HeLa and the red line is the HDF cells. And while there is a section where there is some variability and it looks like HDF is fitting better than the HeLa cells, there just isn't enough variation for it to have any significance, which means that this would not be a viable treatment, at least in the way that we did the experiment, for this to be a viable treatment that didn't damage regular cells enough. This experiment showed that, like I said, again, the dandelion extract being used in this manner to treat cancer cells is not a viable option and couldn't be used in a clinical setting. However, this is not a bad thing. Learning that something doesn't work in science can be just as valuable as learning that it did work. And also, this research will continue past this class using different mixtures. So instead of using that DMEM, we're looking into using, distilled water and other things to mix, and instead of using that chemical specifically. And then also using different extracts from different parts of the flower. Like we use dandelion seed extract. However, you can use things like dandelion whole extract we take from the whole plant. You can use just the stem, the leaves, just the petals. And if this research is successful in showing any variation of this method having an impact on cancer cells with having minimal impact on healthy cells, this could mean it could have a big impact on the cancer treatment field, as cancer is a huge issue. Excuse me. Cancer is this obviously a huge issue and a huge disease that's causing so many deaths. So any treatment, especially one that involves something so easy to grow, highly accessible to many people, could be very life-changing. I do have some acknowledgements. I worked very closely with Dr. Christina Henderson. She supplies all the supplies for this research and her laboratory. I also had a research assistant helping me with this as well, and her name is Caitlin. And then also I had a statistics graduate student, Paul, help me with the statistical analysis and determining whether or not this research was valid. And here are some of my references. All right, thank you guys so much.